Good day, everyone. This is Erin Trisha T. Sinkle, and today I'm excited to bring you a new lesson. The cell membrane is a dynamic fluid-like barrier that is essential for life. But what gives this membrane its fluid properties? The answer lies in the specialized lipid molecules that make up its structure. In this video, we will explore the fascinating world of membrane lipids. The objectives of this lesson are the following. First, describe the membrane lipids and its structure. Second, understand the classified groups of lipids and their connection to membrane fluidity. Third, discuss the membrane fluidity that is determined by membrane composition and temperature. All living things are made up of cells, which have a defining feature of a membrane that separates the cytoplasm from other cells and the extracellular environment. Membranes also surround specialized compartments inside of cells known as organelles. We have prokaryotic cell and eukaryotic cell. Prokaryotic cell is any unicellular organism that does not contain a membrane-bound nucleus or organelles. An example of these is bacteria and archaea. While a eukaryotic cell is any cell that contains a clearly defined nucleus and membrane-bound organelles. Examples of these are animal, plant, fungi, and protist cells. Our discussion of membranes will introduce a third category of biological molecules, lipids. Lipids are a group of naturally occurring molecules that are mainly soluble in nonpolar organic solvents. They are classified into two groups: hydrophobic lipids, such as triglycerides, fats, and amphipathic lipids such as fatty acids, phospholipids, and cholesterol. Lipids are derived from carbon-containing building blocks that are assembled enzymatically by protein complexes in the cell. Now, we will talk about something known as the fluid mosaic model. It describes the structure of the cell membrane as a fluid-like structure with freely moving phospholipids and proteins. This allows the membrane to be selectively permeable, regulating what enters and exits the cell. We will now proceed to phospholipids. The term mosaic used in the cell membrane implies that it is made up of several different components, including phospholipids, proteins, glycoproteins, and glycolipids. Of these components, phospholipids make up the majority of the membrane. Biological membranes are made up of two layers of phospholipids, which form a lipid bilayer. Phospholipids are made up of a polar head group, two non-polar fatty acid tails, and a glycerol molecule. The head group contains a phosphate group facing outward towards the water in both the extracellular fluid and intracellular space, which is hydrophilic and interacts well with water, while the fatty acid tails are hydrophobic and do not mix well with water, creating the interior of the bilayer. Each layer is called a leaflet. So in phospholipids, the polar heads are facing towards the water, and the nonpolar tails are facing away from the water in the interior of the cell membrane. Earlier, you have heard about the word amphipathic. Amphipathic is a term that describes the unique dual polarity nature of phospholipids. These molecules have a polar hydrophilic head and two nonpolar hydrophobic tails, which means they have both hydrophobic and hydrophilic parts. This amphipathic structure is essential to the formation of cell membranes, which are made up of layers of phospholipids. Let us now move on to intracellular and extracellular fluid. The phosphate groups are also attracted to the extracellular fluid. Extracellular fluid ECF is the fluid environment outside the enclosure of the cell membrane. It surrounds all cells in the body. Extracellular fluid has two primary constituents: the fluid component of the blood, called plasma, and the interstitial fluid, IF, that surrounds all cells not in the blood. Otherwise, the intracellular fluid ICF compartment is the system that includes all fluid enclosed in cells by their plasma membranes. Since the lipid tails are hydrophobic, they meet in the inner region of the membrane. ICFs are cytosol, water, dissolved electrolytes, and proteins. Next is the fatty acid. I have said earlier that a phospholipid is made up of two fatty acids, nonpolar tails. Fatty acids are composed of carboxyl groups with long hydrophobic tails or chains that have an even number of carbon atoms and consist only of carbon-carbon and carbon-hydrogen bonds. If the chain contains one or more double bonds, the fatty acid is said to be unsaturated. Conversely, if none of the carbons is linked with a double bond, the chain is saturated. The double bonds in unsaturated fatty acids usually have a cis configuration, meaning that the two hydrogens adjacent to the double bond project from the same side of the double bond. Cis double bonds cause the chain to kink. This is how the saturated fatty acid structure looks like. The two fatty acid chains are part of the phospholipids, which is the hydrophobic part of the molecule. The third part is the phosphate. The phosphate group is attached to two carbons, which is also attached to a nitrogen atom with three other methyl groups on it. The part of phosphate group is the head, is going to be attracted to water. Let us now proceed to the membrane fluidity. How closely packed the phospholipids are. Take a look. If membrane is less permeable, a lot of substances will have difficulty to pass through. If it is become more rigid, it will snap or break. On the other hand, if it is too permeable, different substances will just pass through, and the hydrophobic interactions will be too weak. It will cause breaking. The fluidity of a membrane, or the extent to which the membrane components are free to move, is affected by three factors: the temperature, type of fatty acids, and cholesterol. Membranes can undergo a phase transition in response to temperature changes. The fluidity of a membrane increases as temperature increases. At low temperatures, the lipids within the bilayer are well ordered and packed into a crystal-like arrangement in which the immobilized lipids form strong interactions with one another. As the temperature increases, these interactions are broken, and the lipids become less ordered and more liquid-like. A membrane composed of completely unsaturated lipids will be very loose and fluid, and a completely saturated lipids will be dense and less fluid. This is because longer fatty acids have more surface area with which they can interact with one another. The degree to which fatty acid chains are saturated also affects membrane fluidity. The cis double bonds found in unsaturated fatty acids create kinks in the fatty acid chain that prevent lipids in the membrane from packing together tightly, thus decreasing the surface area over which lipids interact. As a consequence, membranes that are rich in unsaturated fatty acids are more fluid than membranes that contain fewer unsaturated fatty acids. Olive oil and candle wax are familiar examples of this. Olive oil is rich in unsaturated fatty acids such as oleic acid, whereas wax is rich in saturated fatty acids, specifically the stearic acid. As you know, olive oil is a liquid at room temperature, whereas wax is a solid. Let us now move on to the third factor that affects the membrane fluidity. Cholesterol can be found in the membrane. It belongs to a class of lipids called steroids, which are characterized by four fused carbon rings. Not only is cholesterol a major component of membranes, it is also the precursor to other steroids that function as hormones, such as the sex hormones, testosterone, and estrogen, and corticosteroids, which regulate inflammation and other physiological processes. Like phospholipids, cholesterol is amphipathic. It inserts into the membrane bilayer with its polar head group exposed to the aqueous solution and its nonpolar component concealed within the hydrophobic interior of the bilayer. When the temperature goes down, the membrane will become rigid. The phospholipids will move closer to each other, so the fluidity of the membrane decreases. Cholesterol acts to increase the fluidity so as these phospholipids move close to each other under colder conditions. Cholesterol acts as a spacer, keeping them pushed apart. Now, when the temperature rises, the membrane can become too fluid, so cholesterol acts in such way to decrease the fluidity. The phospholipids gain kinetic energy, causing it to move apart, and cholesterol attracts them, causing them to combust. The purpose of cholesterol on the cell membrane is to maintain the fluidity. It prevents it from being too rigid, and it keeps it from being too fluid.